Okay, welcome everybody to the One World Cognitive Psychology Seminar. It's my pleasure to welcome you here today, and especially to welcome Julia Haaf, who I just learned is actually from Mannheim, so welcome to Mannheim. But she will be speaking to us today from Amsterdam, and we very much look forward to that, and thank you that you're taking the time to be here today. So for those who don't know me yet, I'm Beatrice Kuhlmann. I'm one of the organizers of this seminar series, and I'm a professor of cognitive psychology at the University of Mannheim in Germany, and I'll be moderating today's session. And I want to start us off with a few words about Julia, and while I do so, I'll stop my screen share so that Julia can start getting ready for her presentation. So Julia completed her PhD in quantitative psychology in 2018 at the University of Missouri, where she was working with Jeff Rauter. She then completed a postdoc in E.J. Wagenmacher's lab at the University of Amsterdam, and there quickly was promoted to assistant professor. So within less than 2 years, that's quite impressive. Julia's research focuses on Bayesian hierarchical modeling of cognitive phenomena. You might know her work on the Stroop effect as everybody's Stroop. She's also worked on recognition, memory, and subliminal priming. And so she's particularly interested in her research to see how statistical methods can serve us as cognitive psychologists to optimally test our hypotheses. And this is also, I think, something she will focus on today in her presentation. So what can we do with our data to actually measure the cognitive things that we're interested in measuring, particular in disentangling stable qualitative individual differences from mere sample noise. Julia is tremendously productive in her research. Over the past few years, she has constantly published in the best journals of our field, including Nature's Economic Bulletin and Review, Psychological Methods, Journal of Mathematical Psychology, and many more. I think to me personally, Julia is also a role model for open science practices. So she shares all her data, codes, and so on. So if you're interested in that, you can check out her webpage and you can find a lot of useful code on there. So Julia, thanks again also for being here today and taking the time to present to us. And I look forward to learning more about how we can use order constraints in our research to answer our questions.
Thank you. Yeah, thank you so much for inviting me, Beatrice. And I think this is really great. I already gave a talk 2 weeks ago at a different university, also from my house. And it's really nice that we can do this now with modern technology, even in times of a global pandemic, that we can meet up and discuss research. And so I'm really excited to be here and talk about my research. Yeah, so I want to start off by talking about the two disciplines of scientific psychology. And this is a term that was not coined by me. In 2 slides, I'm going to reveal who coined this for those who can't already guess it or haven't read it. And so especially how these two disciplines relate to individual differences. And one of these disciplines or traditions is the experimental tradition, where we develop experiments to understand how experimental manipulations affect behavior. And we typically look at overall effects, right, across participants. And so in the experimental tradition, which you could also call the psychonomics. So this is the study of the lawfulness of psychology or our behavior and thinking and so on. And here we kind of treat individual differences as sample noise, and we try to reduce this noise, right? So we care about overall effects. Individual differences are not really our concern, at least traditionally. And on the other hand, in the correlational tradition, where we use psychometrics, we highlight and value individual differences, and we try to increase it as much as possible. So here, individual differences or individual variability is the signal and not the noise. And so there is a bit of a tension between these two disciplines when we look at individual differences. And actually, Kronbach, who wrote the article, The Two Disciplines of Scientific Psychology, has a really nice quote that says, correlational psychology studies only variance among organisms. Experimental psychology studies only variance among treatments. And a united discipline of psychology will study both of these. And so this tension between the two different disciplines or different traditions is something that we see in a lot of different fields or areas within psychology. And one of them that I'm going to focus on for at least part of my talk is the field of cognitive inhibition. So here we use these behavioral tasks that are developed in the experimental tradition, like the Stroop task or the stop signal task or the Erickson flanker task, to actually understand individual differences of inhibition and how these individual differences relate to other fields. So how do we inhibit irrelevant information, and how does that inhibition performance relate to our working memory capacity, how well we do in school, cognitive development, all kinds of different areas that really relate to this issue. And so here we have a clash of the psychometrics and the psychonomics, or the correlational and the experimental traditions, and we can look what happens, right? And so I'm going to give you a couple of examples from these different traditions and how they treat cognitive inhibition. So the first one, the psychonomic treatment, is really the classical treatment in inhibition research. And so we can go back to, for example, the original Stroop task publication in 1935. This is a citation that almost always shows up in my papers. It's a really great paper, by the way. And it's a very classical experimental approach. It's kind of nice. So when you look at the experiments that Stroop ran, he collected data from 100 participants. So this is, our typical psych experiment. We still do this, like, let's aim for 100 participants. And then, so this example here, this experiment, people looked at either a square with a color and they had to identify the color, or they looked at a word that was a color word and that had a different ink color. So it's like the word red written in blue. And then they had to say blue because the ink color was blue. They had to identify the ink color. And so you can see in this table the response. This is not the response time. This is the mean time for naming 100 colors. We are in 1935. We're not as good as measuring the milliseconds, but we can measure how long does it take people to name 100 of these, right? So this is naming the color of a color word, 110 milliseconds, and then seconds, sorry. And then here's naming the color of a square. That's 63 seconds for 100 of them. And then our effect here is 47 seconds, right? And you could even calculate some sort of effect size measure if you wanted to. But this is this typical psychonomic treatment. So we're looking at the average across 100 participants, a difference between two conditions, two experimental conditions, and this is our result. That's what we care about in experimental psychology. Before I go to the psychometric treatment, there's a bit of an in-between here that is also relevant, and that is the psychophysical treatment. So this is a pretty recent example of that in inhibition, and it's actually, so it's 2 months old. It's actually using the Muller-Lyer illusion as a conflict paradigm. And so what that means is, so the Muller-Lyer illusion is this one, right? It's the one with the arrows. So if you have the arrows pointing outward, the line seems shorter than if the arrows point inward. And so in both of these cases, the upper line is longer. And so the idea is people have to identify the line, the upper or the lower line, which one's longer, right? And so this one is one trial and this one is one trial. And so this one here is one where the upper line is longer and also seems longer, even longer because of this illusion. And in this case, the upper line is longer, but it seems a bit shorter because of this illusion. And so this one you could call a congruent item or a congruent trial. And this one here is an incongruent trial. And if you vary the length of these lines relative to each other, you can kind of get a curve of responses where people are most accurate if the upper line is much, much longer, right? But if they are close, then this illusion will actually have an effect. And so looking at the results of the study, I'm not going to go into detail about them, but what's just to understand the psychophysical treatment, what we have here is the results for each observation, for each participant. And we have 8 of these, right? We have lots and lots of trials per participant and only 8 participants. And in the psychophysical treatment, we don't really care about averaging these necessarily, and we don't really care about the individual differences necessarily. We're caring about the lawfulness. So in a sense, this is still a psychonomic treatment, but we would be able to actually look at individual differences as well, right? And so each panel here is one participant. These are the response time effects, and these are the accuracy effects. And the major inhibition effect is the difference between these two curves for each of these accuracy for each of these participants. And as you can see, this participant has a huge effect, right? And this one has a small effect. So we could actually go through all of these and figure out who has the biggest effect and who has the smallest. If we really wanted to, we could correlate their effect with other things, even though with 8 observations, that's a bit tricky, but it's kind of an in-between. We can identify individual differences, but we still care about the lawfulness. So now that brings me to the psychometric treatment. And I will not give you an example from a recent paper, even though there are lots and lots of them, but I just want to show you what's the typical treatment. What do we typically do in these studies? And so what we have is, the first thing we have is we have several tasks. That's the big difference between the psychonomic and the psychometric treatment. So we have raw data in the green table, which is basically the response time or the accuracy per trial, per condition, per person, per task, right? So this is like long form when you think back to stats or SPSS or whatever, whenever you did analysis in your courses. So this is long format. And then you use this raw data to get performance scores per task and participant. That's the blue table. So one way of doing this is you summarize, like you get the Stroop effect would be a performance score. So this is the average response time in the incongruent condition minus the average response time in the congruent condition. And you get that for each person and each task. And then what you can do is you can correlate these, right? So you can correlate the task performance, and you get the correlation table or the variance covariance table. And then you can plug on this structural equation model or a factor model or some sort of other structure model. So this is where then we try to understand what's the structure across tasks. This is basically just a function of this orange table. It doesn't really have anything to do with the green table. Now, the problem with this current psychometric treatment is that it really doesn't work. So, so far, we've not been able to find a reliable latent structure of inhibition. And if you look at some of these publications that look at these and you look at the correlation table, you find that the correlations are incredibly low. And that is especially the case when you consider how similar some of these tasks are. Like you can correlate 2 different Stroop tasks and you will find a low correlation. And it's really incredible how that happens, right? So nobody really knows yet what's going on. There's a lot of discussion in the field. There are a lot of different suggestions what potential reasons could be. Part of it is like, well, I think the general one is just the tasks are super unreliable, or maybe there is no inhibition, right? Maybe we have to abandon this idea that there is a unified construct of inhibition and task performance shouldn't be correlated. And I, for now, want to focus on 2 potential reasons. One of them is a lack of theoretical understanding of individual differences in these tasks. So if we don't really understand why people vary, then we may not find correlations. And then the second one is that there's no separation of sample noise and true effects. What we are correlating might be very noisy, and if that's the case, that also is related to reliability, of course, then you get low correlations. That just makes sense, right? So in this talk, I'm going to focus on these 2 potential reasons, and both of them can be answered by just looking at one task. And so I'm not going to look at the correlation matrix or correlations across tasks for today. I'll just focus on one task. And because it's my favorite one, I'm going to look at the Stroop effect and the Stroop task. Okay, so let's take a closer look at the Stroop effect. I already said that the Stroop effect is defined as the average response time in the incongruent condition minus the congruent condition per participant, right? And so the typical Stroop effect that we would expect is around 50 milliseconds, maybe. That's you know, if you want to guess, you would guess something in that area. So this is like the usual Stroop effect. And most importantly, this one is a positive effect. So we expect that people are slower in the incongruent condition than in the congruent condition. The 50 milliseconds is not theoretically motivated. It's just my experience of looking at Stroop tasks. I wouldn't be incredibly surprised if I found something like 20 milliseconds or 90 milliseconds. I don't really care about that, but the direction does make a difference. And typically what we do if we, analyze a Stroop task or something like that is we would run a t-test and we would compare it with the idea that there's no Stroop effect, right? So we test, is there an effect versus there's no effect? Or is there a positive effect versus there's no effect? That would be a one-sided t-test. And then there's also the possibility of an opposite Stroop effect or a Stroop pathology, or, something's going on for sure. So that would mean that the people are slower in the congruent condition than in the incongruent condition. I'm not entirely sure how that would be possible from a theoretical point of view. So one idea would be maybe inhibition facilitates responses, which is an interesting suggestion. It's not typically what we find, that's why it's the opposite Stroop, but it's also theoretically somewhat implausible. And so these directions, the directionality of the Stroop effect also translates to individual differences. And so what you can do if you consider the directionality is differentiate between quantitative and qualitative individual differences. So this here would be two pairs, the two observations, A1 and A2. These are two participants, and they both have a positive Stroop effect. And so they vary in their effect, but both of them have an effect in the same direction. So both have an expected Stroop effect. This pair, on the other hand, is a bit different. So here, one participant has a negative effect and one participant has a positive effect. And what's really important here is that the average effect in both of these cases would be positive. So from the psychonomic perspective, we wouldn't distinguish between these two. This is the same thing, right? From a psychometric perspective, maybe not so much, but actually the difference between them is also the same. So maybe you wouldn't care about that either. But from a theoretical perspective, it's completely different, right? Because in this case here, B1 is a Stroop pathology. And so something's going wrong here. Inhibition seems to be facilitating the response of B1, but not of B2. And so in the A scenario here would be quantitative individual differences, implying that they differ in size of the effect, but they don't differ in the direction, or this would suggest a common processing architecture or something like that. And then the B scenario is one where we find qualitative individual differences. The effect is qualitatively different between the people in the set. And so this almost leads to this question that does everyone Stroop that Beatrice already mentioned, but there's still one aspect missing, and that is the issue with the sample noise. So before we start modeling this idea of individual differences, we also need to consider the effect of sample noise on the setting. So what we get if we run a Stroop experiment is we get observed effects. This is the orange points here in this figure. And the observed effect is a function of the true effect and sample noise. So this is what we would observe if we had a limited number of trials, which is every time, right? The true effect is what we would obtain if we had an unlimited number of trials per person and condition. So the more trials we run, the closer we get to that true effect, which is why you should collect a lot of data if you can, per person, if you want to look at individual differences. But we never get there. And so every time we get these effects and we have these repeated measure scenarios, it makes sense to think about sample noise and to model it, to account for it so that we can make hypotheses about true effects, which is what we really care about, because that is really the underlying processing, right? The sample noise is just added noise from people being slow in one trial and faster in another. This is really not what we care about, at least not in this setting. So we're trying to get to these green points, and we actually also want to make hypotheses about them, not about the orange ones. And what's really important about sample noise is it can go in each direction, right? So it may be, like in this case here, that we have quantitative individual differences from the true effects. But after adding sample noise, you smear that out and you add sample noise sometimes so that people's effects become negative. So it looks like we have qualitative individual differences, but if you account for sample noise, they become quantitative individual differences. And so now we can form the research question really, and that is, does everyone show a true effect in the same direction? Or are there some people that truly show an opposite effect? So does everyone stroop, or are there some people who stroop opposite? And one thing I also want to add is, well, is that even an interesting question? That was really the first thing that we thought about when we thought about this idea. It was like, oh my God, stochastic dominance, let's take a look at this. This is really interesting. But then the question is, well, is that really interesting? Or, when you look at observed effects, if everyone already has a positive effect, then maybe this is not that relevant, right? Because we already know. But when you look at observed data, it does get interesting. So this is data from 6 different tasks, 6 different experiments from different labs, different number of participants, different settings. And in 5 of these, you see, so what I did is I just plotted the observed effects and their confidence intervals from the smallest to the largest effect for each of the tasks. And all the pink pluses here, they are negative observed effects. So in at least 5 of these 6 tasks, we find that some people have negative observed effects. So just from looking at observed effects, we would conclude there are qualitative individual differences. And our question is, does that hold if we account for sample noise? And how can we test this? And so there are 3 theoretical positions that we want to look at, and they correspond to the expected Stroop effect, the null, and the opposite, or at least some of them is opposite, but then adding the individual differences part. So the null model would be that no one shows a true Stroop effect. Everyone has a null effect. The positive effects model is everyone shows a true Stroop effect in the same direction. And the unconstrained model is that true Stroop effects vary in direction and size. So they can also be opposite of the expected Stroop effect. And so let's take a look at this in terms of models of 2 participants. It's really tricky to show 57 or 265 true Stroop effects in a figure. So I'm going to go for 2 of them. So on the x-axis, you see the true effect of participant 1. And on the y-axis, there's the true effect of participant 2. And then we can place models in this space. So the null model is really just a point. It means that the true effects have to be 0 for both participants. The positive effects model looks like this. It's kind of interesting. So it's limited to the upper right quadrant, which is all positive effects. And then you also have this induced correlation between them. So if you look at this, it's more plausible. So the darker the area, the more plausibility for that point. And it's more plausible that the effects are relatively similar. And the reason is, this is a hierarchical model. And so the hierarchical structure introduces a correlation between the effects. And it also does that in the unconstrained model. So here we don't have a restriction in space. So the effects can be anywhere, but we again have that correlation that is induced by the hierarchical structure. Here are the 3 models again. So these are the models on true effects that we're considering. And the nice thing about Bayesian modeling, which is what I do, is that we can have these models and we put priors on things. And because we do that, we can actually get predictions of data before seeing any data, which is really, really nice. And so this is what we can do. So these are going from the predictions of true effects to the predictions of observed effects. So this is actually predictions for the data that we observe. And all we do between the true effects and the observed effects is adding sample noise, so smearing out the picture. And this is really the easiest to see for the null model, because we're going from a point to this blob, which means that we expect small effects around 0 for both participants. And then if you think about this in an n-dimensional space, so for each participant, the effect should be relatively small, somewhere around 0. The positive effects prediction looks like this. And so that means that we can find some negative small effects. This is expected based on this positive effects model. So that already looks like maybe this model has a shot when we think back to the data that we saw from the 6 tasks. And then here's the prediction from the unconstrained model, which looks very similar to the model on true effects, except that it's less correlated. So what sample noise does is it takes out a bit of that correlation. And what we can do now is observe data, right? So the ping point here maybe is like observe data for 2 participants, and we can draw them in here. And then we can compare the density at the point of each of these models, of each of these predictions. And if we compare any 2 of them, that is the relative predictive accuracy between these 2 models. And that is the Bayes factor between the 2 models. So the relative predictive accuracy is the Bayes factor. And basically, if we only had 2 participants, we could plot this, right? And then have a really nice illustration of the Bayes factor. This gets really tricky if you have 264 dimensions. Again, the plot was really ugly. And also, it's probably nicer to use a bit better algorithms to actually calculate the Bayes factor than just looking at the plot. But this is an illustration of what you would do for 2 participants. And so in this case here, the data are 2 relatively large positive effects, which means the null model is doing really badly, right? There's no density here, or almost none. The positive effects model is doing pretty good. The unconstrained model is doing pretty good. But because the positive effects model only predicted in this area, and the unconstrained model also predicted in this area, for example, the positive effects model would probably be preferred over the unconstrained model in this case. Okay, so now let's go back to data. This here is one of the tasks that I showed you before. And the pluses are again the observed effects, and the points are the estimates from the unconstrained model. And what happens is we have hierarchical shrinkage, and the estimates are all positive. So this is already kind of a good sign for the positive effects model, indicating that after accounting for sample noise, we get rid of these negative effects, at least for this task. And so here are the estimates for all of these 6 tasks, and you can see that in none of them, these are all posterior means for each individual, and for none of them they are negative in none of the tasks. But before I go and celebrate, I still want to do Bayesian model comparisons. So I'm still going to look at the Bayes factors. And so here are the 6 tasks. We have the number of participants, the number of trials, and here are the overall effects between 12 milliseconds and 90 milliseconds. And now here's the Bayes factor between the positive effects model and the null model. And all of them are in favor of the positive effects model. And if I take a look at the Bayes factor between the positive effects and the unconstrained model, the picture is a bit different. So it seems like there are effects in these that was to be expected. But then in 4 of the 6 tasks, we find evidence for the everyone Stroop's model. So everyone has an effect in the same direction. And in 2 of them, we find evidence against this. One thing I also want to add is the Bayes factors here are much smaller than the Bayes factors in comparison to the null model. One of the reasons is that the null model and the positive effects model make very different predictions. But the positive effects model and the unconstrained model make pretty similar predictions. There's just a bit of a reduction in the size of the space, right? And so actually what I found is that with the priors, I used the maximum of the Bayes factor is about 8:1. So a Bayes factor of 6:1 is already quite impressive. That means that the data must be very much in line with the everyone Stroop's model. But the first thing I want to do when I look at these Bayes factors here and these 6 tasks is I want to know what is going on in these 2. So where are these qualitative individual differences coming from? So I'm going to focus on one of the experiments here, and I'll show you where I think the qualitative individual differences are coming from. So here are the data again. The pluses are again the observed effects, and then the estimates are here. This is the task with the smallest overall effect, which is notable. And there is some evidence for the unconstrained model, even though this is not very impressive, right? And so what is the task? Well, it's a location Stroop task, which means what the participants did is they had to focus first in the middle, and then a word would appear either on the right or on the left side of the screen, and it was either the word right or the word left, and they had to identify the direction, not the word itself. So in this case, the correct answer would have been right. This is a congruent trial. Here, the correct answer is left. This is an incongruent trial. Then again, right is the correct answer, again, an incongruent trial. And oh, I think I didn't have the one, the last one. But what is important here, if I'm going to go through these again, maybe you can notice this too. So people could make a really good, or it would be a pretty good strategy to just focus on the middle, try to blur out the words, and then try to respond. Because you can still see that a word is appearing in the periphery, but you don't have to read the word at all, right? And so then the interference or the inhibition problem would go away because you don't have to read the word. And so what is possible is that different participants use different strategies. So if I'm going to go back to the data and you look at the observed effects here, so my guess is that there's a bunch of people who just looked at the fixation cross and then responded. And so they don't have an inhibition effect. And then there are a bunch of people who actually attended to the words and then they got an inhibition effect. And so we applied a model that is like a some do, some don't model. This is a mixture model where participants are classified in either a category that has no effect or a category that has a positive effect. And the nice thing is also you can use that classification later, so you get a posterior classification. That model was actually preferred over the unconstrained model. So we found evidence that some people show an effect and some don't. And here are these classifications. So the darkness of the point is the posterior probability that that person has a positive effect. And that probability was between 0.2 for the people with the lightest color to 0.99 for these people. So it seems like this was actually the case. These people here are our nose troopers, and then these people here are the people who actually attended to the words. And one thing I also want to note is that what Pratt et al. Did in the second experiment after this one happened is they put in stop trials into their Stroop task. And so in that case, participants had to attend to the word, and if it said stop, they were supposed to not respond. And that increased that Stroop effect by a lot. They only looked at the overall Stroop effect, but still, it seems like this was really what's going on. There was some sort of strategy use going on in the task. And so looking back at this understanding of the Stroop task, everyone's Stroops versus some do, some don't, versus the unconstrained model where people actually have opposite effects. This has implications for inhibition and also for understanding individual differences and for the correlations. And this is what could go on in at least some of these tasks. So if you have qualitative differences, the distribution of tasks of performance scores might look like this, right? So we have like a normal distribution, the overall Stroop effect, say 50 milliseconds. Some people have a negative effect, some people have a positive effect. And the individual differences, the variance of this curve can be independent of the mean. So that means it doesn't really matter where the overall effect is and whether that effect is small or big, because people might still vary as much as they would if the effect was bigger. If we have only quantitative individual differences, that means the distribution is bounded at 0, right? So it could, for example, be a truncated normal, but it could also be a gamma distribution. It could be whatever you want. In any case, with a bounded distribution, there's a dependency between the mean and the variance, which means if you have a small effect, like we typically have in these inhibition tasks, right? 50 milliseconds is not a huge effect. Then the variance is also bounded. And that means people just don't differ a lot, right? And additionally, they might not differ in the way that we care about, right? If there are no qualitative processing differences between people, then maybe the difference doesn't mean as much. And so correlations won't occur because being good in this task may just be a function of whether, I don't know, you are good at distinguishing between colors or not. And then that doesn't translate to a Simon effect or something like that. And so there is a real dependency between the difference between quantitative and qualitative individual differences and the correlations that we find within the tasks. All right. So this was the last I wanted to say about Stroop effects and the Stroop task, but I also want to show a couple of other applications of this understanding of quantitative and qualitative differences. And one of these applications is in psycholinguistics, and this is a project I'm currently working on, so I don't have a lot of answers yet, but I'm very excited about it, and that's why I want to share it. And so I don't know how much you know about this, so I'm going to talk a little bit about what quantifier use is. So the idea is, how do people interpret quantifiers? And quantifiers in psycholinguistics are things like more than half, or most, or less than half, or few, or many. So there are different quantifiers. And so the idea is, let's say you have a display like this one below here with orange and bluish points. And then I'm going to give you a sentence like, more than half of the points are orange. Or most of the points are orange. And you have to indicate whether you think this is true or this is false. So what I want you to do is put in the chat, I don't know, I think people can use the chat, right? Put in the chat first, whether you think it's true or false that more than half of the points are orange, whether you think that most of the points are orange, and maybe also guess on how many percent of the points are actually orange, just so that we have an idea of the differences. Okay.
So you want us to answer all questions?
Yes.
Okay.
Or as many as you want. Yeah, more than half, most, and what's your guess? All right. There are a lot of trues. But some people have a difference between true, more than half, and most. And I think most people guessed between 55 and 70% of the points are orange. I think it's actually 55, 55% of them. And so what happened was, I was in a lab meeting and a grad student presented this data from the study that she ran. So she did something like this. She gave people these sentences, and then she asked them, is this true or false? And what she found repeatedly was that people interpret most as more than half, which completely blew my mind, by the way, because for me, this is just the same thing. But apparently it's not for everyone. And this is where the question's coming from. How do people interpret these quantifiers? And is a quantifier the same for each person? So if we communicate these things, do we all understand the same thing if we use a quantifier or not? And so the question that we started out with was, is most more than half for everyone? Or are there some people who most is less than more than half, or is it the same for some people? So we just wanted to see what are the individual differences in quantifier use, and how can we understand what's going on. So I'm going to start with some data. This is basically a percentage. So people got sentences like 55% of the points are orange, and then they had to indicate whether they, so then they got a sentence that was few points are orange or many points are orange, and they had to indicate whether that's true or false. And so what you see here is the proportion of true responses for each of the percentages and for each of the quantifiers. So if I just give you an example, the more than half one is the pink one. And that one has a really clear cutoff at 50%, which is what we would expect, right? So if it's below 50%, people say false in every trial or almost every trial. And if it's more than 50%, then people say true in almost every trial. So there's a clear cutoff here. It's a bit different for most. There's the same thing happening for below 50%, but above 50%, there seems to be that some people, or in some trials, people indicate false if it's between 50% and say 65%. So there's a bit more of a smoother curve going on here. And then another thing that's kind of fun to look at is whether few is like a mirror of most or not, or many actually. Few or many should be mirrors, and fewer than half and more than half should be mirrors. And so that's also something that we started to look into, but it's not clear, and I don't think I find a lot of indication that that's the case. And so what I did is I basically I used sort of like an IRT model, or it's very similar to an IRT model. So it's a logit model where the quantifiers can vary in both difficulty. So this is like moving the curve around in this direction, and discriminability, which means the slope of the curve can be different across the curves. And then I use the hierarchical model, so each person has their own curve for each of the quantifiers. And so these are the overall curves that you get after looking at the model. And what's really interesting for that one, that was the only thing that we really found super interesting about the overall ones, is that fewer than half seems to have a really low discriminability. So the slope is much less steep than all the other curves, while more than half is going exactly through the 50% and is pretty steep. Fewer than half doesn't look like that at all. So I thought that was pretty interesting. And then if we go back to more than half and most, you can see that there's definitely a difference in the difficulty, right? So it seems like most is a bit more than half, at least on average. And so here are the individual curves as well for each of the quantifiers. And so the individual differences look very different across the quantifiers. So for fewer than half, there are more individual differences in the discriminability than in the difficulty. And by the way, psycholinguists would call discriminability vagueness. So how vague is the quantifier? Do people use it differently in each of the trials? But for few, for example, there's much more individual differences in the difficulty, right? So people actually move their threshold around, or people have different thresholds. Same for most. So more than half has the same threshold across people, right? Almost everyone has this 50%. Most seems to be shifted, right? There are a couple of people who have a much higher threshold than for more than half. All right, so what we also did is look at these differences between most and more than half. So this is what you see here. These are the estimates from the model. And there also is a small correlation between the threshold for most and more than half. This here is, by the way, not on the threshold, not on the percent scale. This is an internal effect. So this is not really interpretable, but it's different between the two parameters. But we don't find evidence for the everyone does model. So it doesn't seem that everyone interprets most as more than half. There seem to be some people, and this actually looks a bit to me like with the Pratt-et-al's true paradigm, maybe some people actually have a no difference and some people do have a difference. This is something we didn't look into yet, but we don't find evidence that everyone interprets them the same way. We also looked at many and more than half. So this is more than half minus many, the difference between the two. So more than half should be higher than many typically, but so the effect should be positive from that expectation. And it is for a lot of the participants, but not for all of them. And again, we don't find evidence that everyone interprets these or orders them the same way. And there's also much more individual differences, individual variability in this, because many is such a vague quantifier, right? People just have very different thresholds for those. And there's also almost no correlation between the two, many and more than half. There's a lot of variability in the threshold for many, but almost no correlation. All right. I don't think I have a lot. Well, I still have a little bit of time. It's nice because I still have another application that I wanted to share with you, which is meta-analysis. This is a bit of a shift, but it really is a nice application of this idea of quantitative versus qualitative differences. But now we're moving from participant differences or individual differences to study differences. So let's start with the usual meta-analytic question, and that is, typically what people ask is, what is the overall effect combined across a bunch of studies, right? We want to know, what is the effect size estimate across studies? The argument is this is a good idea to look at because if we do study planning for future studies, it makes a lot of sense that we can do a good power analysis, or we want to know what's the evidence for an effect or something like that. So people look at this overall effect. And that's this is parallel to the psychonomic approach, right? So we're just looking at the overall effect and ignoring how studies vary. Sometimes we look at whether they vary or not, and then we try to explain that variance, but this is more of a secondary question in most cases. The problem is there can be a lot of reasons why the overall effect is the overall effect it is. And there might be systematic or arbitrary effects, right? The overall effect depends on the choices of paradigms and variables that people use, and also what is hot in the field. So maybe someone came up with a task this way and then other people copied it without thinking whether the number of trials make a lot of sense or whether we should have done something differently. There could also be systematic effects like publication bias, but in any case, there are a lot of reasons why the overall effect ends up what it is that are not theoretically meaningful at all. So they are just a function of what people do as researchers and not, the psychological effect itself. And so maybe focusing so much on this overall effect isn't really that interesting because, sometimes you see that these effects can vary drastically across studies, and it's just a function of what's the proportion of this type of study being in your meta-analysis versus this type of study. And that's arbitrary. There's nothing, this is not like sampling like we typically do for participants. And so it just doesn't make any sense to focus on that so much. And so what we thought was that if the target contrast is robust, then the direction of the effect should not be affected. So no matter what the overall effect is, we should find the effect in the same direction in all of the studies, right? So it doesn't matter why they vary and how they vary, we just want to know what is the prevalence or what is the evidence that they are all in the same direction. So here we are back at the ordinal constraints. And so this is also how we can use them in meta-analysis. I want to highlight that Jeff, I think he's here, I saw his name in the chat, so hi Jeff, wrote a paper on this together with Clint and Joe Hilgard and me, and we also just submitted a preprint on this, and the question that we are asking is, does every study in a collection plausibly show an effect in the same direction? This is also not necessarily a completely new idea. There's actually a reference from the 80s already that looked at qualitative interactions. And this has to do with in clinical research with treatments and how they affect different participant groups. So it might be that one treatment is better for one population, but another treatment is better for another population. And then we would find these qualitative interactions where the treatment effect between treatment A and B is positive in one population and negative in another one. And that's obviously something we would want to know, because then we would make different clinical recommendations than if everyone prefers treatment A, right? So this has to do with the idea of generalizability versus qualitative interactions. And so we would say that if every study in a collection shows an effect in the same direction, then that means that this is a general and generalizable effect. And if not, then we need to understand why not and what causes these qualitative interactions. And the models that we can use here look very similar to the ones that I showed you before for the Stroop effect, right? So we again have effects for each study. Study 1 is on the x-axis and study 2 is on the y-axis. And we can basically use the same model with a couple of modifications. One of the problems is we typically don't have raw data available. So we need to model the effect sizes, the effect size estimates. But the idea is pretty much the same. And we look at the null model versus the positive effects model, meaning every study shows an effect in the same direction, versus an unconstrained model where that ordinal constraint is removed. All right, so let me show you an example real briefly. And this is one from developmental research, and it has to do with early language development. Carbajal and colleagues conducted a meta-analysis on this, and they looked at whether toddlers recognize familiar words. So the task typically looks like this. You put a baby in a room, and then you have different speakers in different directions, and you either present a word that is familiar to the toddlers or a word that is unfamiliar. And the idea is how long they look in that direction where that word was coming from is a function of whether the word is familiar or not. And so in this age, the hypothesis is that toddlers pay longer attention to familiar words than novel words, so they would look longer in the direction when the word was familiar than when it wasn't. So we have this meta-analytic data set with 34 studies. Here are the data. The squares are the observed effects, and the points are the estimates from our unconstrained model. And so as you can see, you also get shrinkage, by the way, in meta-analysis. So this is what you can see here. And one thing I also want to point out is this is using the effect size estimate Fisher's Z. For experimental psychologists, this is something that we are not super familiar with, or at least I wasn't. The interpretation is, or you can scale this to Cohen's D, and Cohen's D is usually about twice of Fisher's Z. So even though both are standardized effect size estimates, they're not the same. They're not on the same scale. Fisher's Z has a different scale than Cohen's D. And so when we look at the Bayes factor model comparison for this familiar word recognition data set, we find that the every study does model is preferred over the unconstrained model by about 15-1. So there's evidence that every study shows an effect in the same direction. Let's go back to the data. From the observed effect sizes, there were actually 3 of them were negative. But if you look at the estimates, all of them are positive. And so we have this evidence that every study shows an effect in the same direction. And the Bayes factor of the every study does model over the null model is about 9-1. So there's evidence for that as well, which means we have evidence that every study shows a familiar word effect. So there seems to be this effect. And what's nice about this is now that we have this evidence, we can at least tentatively also interpret the overall effect, right? At least we know that the overall effect to some degree describes all the studies in our data set. It may still not be that interesting after answering this question, but it at least represents something for all of these studies. So the overall effect here is about 0.2. And as I said before, Fisher's Z is about half of Cohen's D, so you would say Cohen's D of 0.4. And then what's also interesting is when you think about this question, does every study show an effect in the same direction? Then also accounting for predictors changes, or what we care about with predictors. So we go from what is the effect of the predictor of the average on the average effect size to how does the predictor affect the qualitative nature of the distribution of effect sizes? And so I'm going to show you what that means for continuous and categorical predictors just very briefly. So what we could do, for example, in the familiar word recognition meta-analysis is we could look at the effect of age. So this is the standardized average age for each of the studies that were included in the meta-analysis. And then this is the effect size. And again, we can look at the empty points or the observed effects, and then the estimated effects are estimated, they shrink towards that regression line. This is what typically happens if you add a predictor in this. And so there are a couple of things we could think about when we look at this predictor. So one thing is, well, suppose we found that some studies show an opposite effect, right? We have evidence for the unconstrained model. Then we could care about, well, if we add the predictor, can we account for that variability and then actually all effects become positive, right? So using the predictor to actually move all the effects up, and then we find evidence for or against the qualitative differences between studies. Another possibility is actually the opposite. So if we find evidence that all studies show an effect in the same direction without the predictor, adding the predictor can explain like noise variability, and so the shrinkage pattern could be different. It could actually mean that some studies shrink less because the predictor explains why the effects are negative. And so we could also ask, after accounting for the predictor, are still all the studies in the expected direction? So using a continuous predictor really kind of complicates the pattern, but there are a lot of interesting questions that we could ask that relate to the distribution of the effects. If we look at categorical predictors, this is also super interesting. So this categorical predictor that we used here is basically, was the effect used as a baseline in the study? So because it's such a standard effect, people sometimes use it as a baseline effect and then they actually study something else. So the target effect is something different. So they use it as a baseline, or they use it as the target of study. And so what the original authors of the meta-analysis actually told us was, well, we have this suspicion that if it's a baseline in the study, people will omit it if it's not significant. So they only publish positive effects if it's a baseline effect. If it's the target of study, because developmental data is so costly, people have to study no matter what, they will publish no matter what, they will publish it even if it's not significant. So it might be that we have some sort of publication bias going on where the biggest effect sizes or the most positive effect sizes are from the baseline studies. And then there might be no effect if you look at the target studies. So this is what we see here. The estimates are 0 for all the studies that were target studies. And for the baseline studies, they are actually, so this is again a mixture model like we had for the some do and some don't model. So some studies show no effect and some studies show a positive effect. And if we compare this model to the positive effects model from before, we actually get evidence for this model. So it seems that some studies actually show no effect and some studies show a positive effect. And it's really just a function of whether it's a baseline or a target effect condition, which was a bit of a frustrating thing to see. But I think that the idea of using categorical predictors this way is quite interesting. So you could consider, for example, in learning or in conditioning, we could take a look at, or even better, priming studies, where maybe we find no effect for subliminal priming studies and we find an effect for supraliminal priming studies. So there are a lot of these categorical predictors that could lead to this pattern where for one level of the categorical predictor, we find no effects, and for others, we find an effect. And looking at this, these patterns in meta-analysis, I think is also really interesting. All right. So those were my two applications that I wanted to talk about. So now I still have a bit of a conclusion. So as a takeaway, I think it's really interesting to look at these patterns and to look at quantitative and qualitative individual differences. And the idea is that if everyone shows an effect in the same direction, then we can rely on a simple and universal theoretical account. At least there is a possibility that this is enough. And if some individuals robustly show no effect, this can have implications either for the experimental method or for the theory. So it might be that these are just individual differences in how we process things, but it also could lead us to to thinking of different strategies, right? So in the Stroop example, for example, what happened was that people used different strategies. There was too much flexibility in the experimental design to really look at the processing going on. We basically had this layer of strategies on top of it. And so if you want to understand whether people differ in their processing and they can only use one strategy, then maybe we have to adapt the experimental method. And so this can also lead to a different understanding of the experimental method that we're using. And if some individuals robustly show an opposite effect, then we may also prefer more complex theories, right? So this might point to different processing architectures. There's something fundamentally different going on between participants, and we have to understand why. And in a lot of cases, especially in cognitive psychology, it's really hard to come up with a good example for this as a hypothesis. So a field in which I would hypothesize that this is going on. And so we had a lot of brainstorming sessions on this, and I had a really hard time to come up with this outside of strategy use or preferences or things like that. There it's kind of easy to come up with that. People prefer different things, but if you really think about basic fundamental processing, then it gets really difficult. And so if we think back to these different disciplines, the two different disciplines in psychology, the takeaway from the psychometric perspective is that we should assess how individuals vary without overstating individual differences. So especially when we have these repeated measures designs, we should always use hierarchical modeling and account for sample noise, because otherwise we might overstate individual differences and we might think that there are qualitative differences, even though they really are only quantitative after accounting for sample noise. And from the psychonomic perspective, we need to first know that people have a similar processing architecture before we can really interpret average effects. And this also translates to meta-analysis, but I think it's even more important also in our experiments. So if we don't know whether the average really is representative of all people, if some people have an opposite effect, then the average is really difficult to interpret. It's an average across a mixture of effects and of qualitatively different outcomes. And so I think we really have to understand what is going on there before we report an average effect and just go on with our lives. And so I'm going to finish with my favorite quote from that paper from Kronbach from 1957. And I feel like this is very typical in our field, that someone always said it better before. So this one is really nice. He says, in both applied work and general scientific work, psychology requires combined, not parallel labors from our two historic disciplines. In this common labor, they will almost certainly become one, with a common theory, a common method, and common recommendations for social betterment. In the search for interactions, we will invent new treatment dimensions and discover new dimensions of the organism. We will come to realize that organism and treatment are an inseparable pair and that no psychologist can dismiss one or the other as error variants. All right, well, thank you for coming to my talk and listening, and I'm always open for questions and discussions. So if you have any questions, the chat is open and you can just chime in.
Yeah, thank you, Julia. I'm sure this was interesting to most, if not all, who are here. And yeah, we have the floor open for question. You can also ask questions directly by using the raise your hand feature. So if you hover with your mouse on the bottom, you'll see the menu bar from Zoom, and there's the participant tab, and in there you can find the option to raise your hand. And somebody already found it, so I'll unmute Uli Glovala so you can ask your question.
Can you hear me?
Yes.
Wonderful. Julia, I really liked your talk. It was extremely interesting, and I think we should have more of this in psychology and, let me say, in many other sciences as well. The one problem that I see is that in quite a few studies, I have the impression that we do not have enough data for each individual subject to come up with a reasonable account of the behavior of that subject. In other words, well, I've done a couple of, well, perception experiments where you just have 3 or 4 subjects and you test them extensively, like for weeks. And at some point in time, you start to measure after a training period. So then I really have the impression that these are reliable behavior patterns after such an extensive training. As a matter of fact, at least you want to take 3, because if the 3 are the same in their behavior, then the likelihood that they display different strategies is fairly low. But in many other experiments that many people have done, there is just not enough data for each individual subject. What do you do then, my dear friend?
Yeah, that is a great question. Yeah. So in fact, in these inhibition tasks or these correlation studies are a good example of when we don't have enough data most of the time, because, well, I mean, most people don't have the psychophysics approach. They don't get the people back into the lab over and over again, and right. And so you have to run, say, 10 different tasks. This battery is huge, which means you have to distribute the attention of that participant across all the different tasks, and you end up with very low numbers of trials in each task. And that is a huge problem, right? And we will never be able to disentangle the sample noise in these tasks and the signal. It's just impossible. One of the recommendations that Jeff Rauder and I gave in a paper was that we need much more trials, many more trials within a task to really do these correlational studies. And that obviously also counts, like if you just have one task, you need a lot of trials to really understand the individual differences within the task. I think what the modeling approach that I propose can do then for you is it can highlight that you don't have enough trials, that you can't learn.
Yeah.
Right.
Yeah.
So
That's
even though you can learn, you at least know that you can learn. And if you don't use it, if you just look at observed effects, you might think you're learning.
Perfect. We need more grant money for doing this, you know?
Yeah, that's true.
You should go to the funding agencies and tell them that researchers have to do more trials on each individual task.
Yeah, I recently looked at data from a developmental study. This is a huge conglomerate in the Netherlands across several universities. They look at these longitudinal developmental studies within children, and they look at inhibition, right? So this is like social control. They look at social control and then how does it relate to inhibition. They also use these tasks. They actually use a flanker and a stop signal task, and they have 8 trials for each task in each session. And it's just, I don't know what they're going to do with this data. I don't think they will be able to see anything from it. So I think it's really sad, because it's a big study. It would be really great to have good data from each of these kids, but we will never really know how it relates to their to these behavioral tasks.
Yeah, what a shame. Thank you so much.
Thanks.
Okay. And I see that Arndt has a question, so.
Yes, thank you, Julia, for this inspiring talk. And it's, of course, a problem to estimate whether there are individual effects for each individual. But may I play the devil's advocate a little bit, because your conclusion sounds as if the only solution would be to have more data for each participant to reliably estimate the participant's effect. But this somehow depends also on what you want to do with your study and what your philosophy of science behind it is. So if you have a deductivist idea, a Popperian idea of testing hypotheses, then of course what you do is you have a hypothesis and you make a prediction given some auxiliary assumptions in your experiment. For example, that the experimental groups do not differ a priori. and then you deduce that there should be a difference at the group level. And why not in some situations, if you cannot sample more data to look whether each person shows the effect, you still have valuable evidence then by testing your hypothesis deductively. And you don't want to make stronger claims for the data that cannot support these claims, but still this wouldn't be useless information you get.
Of course not. No, I think there are two aspects in this comment, I would say. So one is this. So what happens if you cannot collect more data, right? And this is also very commonly the case if you look at, for example, social psychology experiments, right, where repeated measures are not an option because this might change the responses. This is obviously also the case in cognitive psychology, where you can have training effects or people get tired. So you cannot, obviously we cannot collect 1,000 trials per person per day, or maybe 1,000 would be possible, but maybe not 10,000. So obviously this is the case. And then what we really have to do is we have to limit our hypotheses and also our conclusions. And I do have the impression that this is not necessarily the case, right? So it's not like our discussion sections or our results sections typically include the sentence, well, there might be individual differences, but we will never know, and we cannot account for them for these and that reasons. And so it is, the average doesn't really describe, might not describe everyone. I've actually never read this anywhere. We obviously sometimes we do write this like there's an overall effect and this is on average, but I'm not sure whether we really interpret it in this way necessarily. And I do think this is really, I think this hypothesis or this idea that makes a lot of sense from the psychonomic perspective if you say that these individual differences are not what you care about, right? So they are noise. They are not what we care about. But I wonder whether that's really the case when you want to make conclusions about processing, right? Because processing is always going to be something that is within a person. And so if we say we don't care about individual differences, I'm not sure whether that makes sense at all.
No, I didn't say I don't care about these individual differences, but if I do not have a chance to estimate them, still I can conclude reasonable things from a deductivist test of a hypothesis. And even if I have no idea how much these results are affected by noise, for example.
But so suppose we have two groups now in your study, right? So you're concluding there's a positive effect, but you have 60% of the participants have a positive effect and 40% have a negative effect. What's the average then?
I don't care about the average. I don't care about the average.
But you're testing the average.
Yes, but I only test the average because I have no better way of testing the underlying hypothesis that there is a difference between the groups, which may be due to each person in the group. So it might be a universal hypothesis which just states everything else equal, ceteris paribus, there must be a difference. And if I ensure this in the experiment, then I have no control over the noise in the sample, but still I can make a prediction that there must be a difference. And if there is this difference, then I corroborate my hypothesis. This is weaker than showing it for each person, but still it's a valid argument, I would say. And it doesn't mean I do not care about the individual differences. I would love to estimate them, but if I do not do, then my study is not useless. And it sounded a little bit as if you said, if we can't do that, then we cannot test universal hypothesis. And I do not agree.
Okay.
Okay, and we have a question by Daniel Heck.
Hello.
Yeah.
Hi. So, hi, Julia. Thanks for the talk. As you might guess, I like this type of work, testing order constraints. Still, I wondered, and this question goes in a similar direction as Arndt's question. Shouldn't we care more about representative sampling once we talk about all people? If you want to say that all people do this or this, you can only say all people in my study did this or this. So suddenly it becomes much more important to take into account who are the participants who participated. You can have 1,000 students who all have an effect, but there's one counterexample of, let's say, an older person for whom you don't find the effect. And then deductively, you can falsify the theory with just one person, but your base factor will be huge to show that for this sample, there's huge evidence that all people do something. So is this a problem we should care more about if we do this type of research, or is it not a problem? Or
no, I agree with you. I think that is a problem, and that's also, it is something that I sometimes found when I looked at these studies. So in inhibition research, it's pretty common to also include older adults in the sample, especially if you look at these correlation studies. And so what there is, I can't quantify this, but I have looked at a couple of these studies, and they did have more individual differences, not necessarily the qualitative individual differences, but at least higher variability. I agree with you that especially if there are hypotheses about a mapping between qualitative differences and say age or other variables, that it makes a lot of sense to think about representativeness. I do think in a lot of cases we don't really have that. So we have a mapping between more extreme effects in age, for example, but we don't necessarily have a mapping with qualitative differences. So I think it would be even more valuable to look at representativeness if there are areas in which there is this hypothesis, so there could be that hypothesis. Yes and no would be my answer.
It's a difficult question, because one very strong conclusion you could draw is, as long as you don't have representativeness, you cannot say anything about the population. Right. Because you have biased sampling, you don't know in what ways, and that's it. And of course, we don't want to do that. We want to test something. And so I think the method is still useful, because it tells you in this specific sample, for whom I do not really know who it is and who they were sampled and so on. But at least in this sample, in this convenience sample, there is evidence or not, but it's a little bit more limited than just saying all.
That's true. So there are two additional things that I want to say. First of all, this would also have an effect on your overall effect, right? So if there really were these mappings and there were these qualitative individual differences, you might find a very different pattern in your overall effect if you sample different populations, right? So this would, for example, something that would show up in meta-analysis. And the second thing I want to say is there's actually one example where there is this mapping between age and qualitative individual differences, and that is in memory of like an interaction between memory and recognition memory and valence. So it seems that older adults remember positive words better, and younger adults remember negative words better. It's actually, so there is evidence for this effect in the literature. I'm not sure how strong this is and whether this would replicate necessarily, or this would also translate to other areas. But I think there's actually, there should, so this is a good example where it really matters who you have in your sample.
Yeah, thanks.
Okay, and we have another question by Uli Glovala.
Can you hear me?
Yeah.
Wonderful. Let me pick up on what Arndt has arguing. I mean, we're not giving up that easily our experimental psychology. I mean, you're saying the way we do experimental psychology is, not optimal. To some extent, I agree, but it's not quite like this, I think. Typically, if you run a series of studies testing hypotheses about very similar processes. This is what you usually find in a publication. You do it with material X and material Y, with the subject group X and Y. You have a couple of manipulations, and well, if all these studies, even though you just compute the means and then calculate some statistics over it and have not enough observations per subject, if you have a number of experiments that all go into the same direction, the likelihood that you aggregate over completely different behavior patterns is not that high anymore. So there is some limits in the strong point you're making by aggregating over a couple of experiments, I believe. If you say, Uli, this is wrong, I'm stuck.
Think about handedness. Say you have a prevalence of someone having of opposite Stroopers, of Stroop pathologies in your population of 10%. And you always sample, and your samples are, great. They always get these 10% in each of your studies. Then average effect is more likely going to be positive every time.
Yeah.
Right? Then, of course, the nice thing about this is you can say, well, I guess at least the majority of people is represented by the average, but it's still, it still would be obviously better for us as experimental psychologists, or for me or for cognitive psychologists who wants to study lawfulness, if that lawfulness is actually representative of everyone.
Yeah, but this is, I mean, this is, I compare that to a religion, a Christian religion. The Catholic Church invented something which is very, very helpful. It allows you to make sins, and they forgive you at some point in time. So you're good again. And what am I trying to say with this analogy? What we are doing right now is probably the best we can. And I think we just won't have the time and the money to get sufficient amount of observations per individual to do it always the way your results suggest. I believe aggregating over a couple of experiments is a pretty good guess that you're close to the truth. And if you say, Uli, this is not true, the world is different, well, this is what I'm used to, then please run a couple of studies and show me that your model fits better. That's the way it is. This is a little side remark to the representative groups of subjects. The subject that you have in a study should be representative for a population. We all know that most of the studies, even studies that we find great, and the people who run these studies are great, they are by no means representative. They're just student populations, and this is true for your university and many other universities. I think this is a problem. Yes, it depends on what you want to generalize. But if you're satisfied with the results so far and do not have strong counter evidence, like the example that you gave, older people behave different in terms of variable A and B. Well, then, of course, you have to modify your claims. But this is the typical way that we proceed. It's not a big deal.
Well, that's a good question whether it is a big deal. I think we can obviously discuss a lot about the practicalities of this approach. But so what I presented and what I'm hoping I'm suggesting is a different approach. This is a method that you can use, right? And so whenever you have enough trials, and actually, so looking back at the 6 Stroop tasks that I modeled, the minimum number of trials per condition was 47, which is not a lot, right? This is not a huge experiment. So this is something as experimental psychologists that we have a lot,
Yeah.
or we have in many experiments. And so if that's the case, then you can apply this method and you can maybe learn more than you would from aggregating. And that's what I'm suggesting. Whether that's practical for all experiments, probably not. But I think that using hierarchical modeling and also considering the ordinal constraints that come from your predictions is something that I would like to see more in experimental psychology.
I perfectly agree. And let me add one little thing. In economics and computer science, people make a lot of out of analyzing big data. And in psychology, I have the impression that we are not yet so far that we dump all the data that we collect in experiments and put them in huge databases described in a reasonable way that we can compute on these big data. Perhaps aggregating over a couple of similar subjects is not that far away from aggregating or collecting whatever 100 trials per subject. You know, if you see a particular pattern of behavior, look at all the people that have this pattern and collect some others that have a different pattern, and then try to find out what's the difference between those two. So I'm trying to say, look at many data. Let's have many data and analyze them. I mean, basically, that's what you did. You took a lot of studies and looked at them. The meta-analysis was wonderful. And we have to learn to do this to become more sophisticated, to make our predictions more precise. That's a good point. This is true for almost each and every science. Look at the virus people right now. I mean, you have very smart people from many different places, and you at least have 35 different opinions, and they are all smart. They are all collecting good data. They all have their strategies, and well, yeah, but you're right. We could do better. I agree.
Thanks.
Julia, you have a good point. You have a real good point. You got me. Thanks a lot.
I'd like to give Martin a chance.
Yes, thanks.
Question.
You can hear me? All right, great. Hi, Julia. Thank you very much for this talk. It was a great talk and very inspiring. And obviously, I'm a great fan of this approach of yours. And I do not want to add to this philosophical discussion. It's a way more practical question, inspired, though, by this discussion, namely on the meta-analysis part. So I think this is a very great question to ask across studies. And my question is, when you use the studies basically as the data, the number of studies becomes your sample size. And my question would be, I mean, when it comes to estimation, of course, the number of samples will affect, for example, the impact that the group or the hierarchical account has, like the effect of shrinkage. What about the group or the model comparison? So if I, for example, want to make the point in a number of studies that there is no effect, I want to compare, for example, my null model to a positive effects model. Would you say there is a number of studies that I should have in order to have a reasonable chance to gain evidence or to see evidence for my models? Or is this very, like, does that vary from case to case?
That's a really good question. I mean, yeah, I would say it probably, yeah, it does. I'm sure it varies. I mean, it really depends on what the effects are, right? I mean, I looked at these 34 studies, which I would say is already relatively small sample size, I guess, of studies, and this worked out pretty well. So my guess is that you can go relatively low if you just care about that question. Then looking at predictors obviously becomes much more tricky, and then you need many more studies. There's actually also one thing that I noticed with this approach. I also analyzed a set with a lot of studies, with over 400 studies. And what happens then is you almost always get overwhelming evidence for the unconstrained model. This is also a function of the priors and all that, but it seems like in such a sample, you just have too many violations, right? You have too many studies that show in the opposite direction to really get evidence for the every study does model. So there's almost like an upper limit, and I haven't really figured out how to scale it appropriately for these big numbers of studies. That's something I still have to take a look at.
All right, thank you.
Okay, thanks again so much, Julia, for this interesting talk and discussion. And we're right on time to give you a final clap and invite everyone else to join us next week. We'll have Norman Brown with us next week, and he'll talk about some current effects of the COVID-19 pandemic, the effects of this pandemic on the contents and organization of autobiographical memory, a transition theory perspective. So see you next week, everyone. Thank you.
Thank you.